Hello, I'm Dennis Mehay. I'll be presenting Catalytic's DrugProt Challenge system submissions. A brief word on our general approach is that we focused only on doing NLP on a per sentence basis. We didn't use knowledge from ontologies, experimental data sets, drug or protein structure, or anything of that sort. We also don't perform named entity resolution across sentences, which would allow us, for example, to propagate the most confident prediction throughout the abstract. Our two systems are as follows. As a strong baseline, we used the BioBERT system of Lee and colleagues, which we re-implemented in PyTorch. This system was competitive on the ChemProt dataset, and so it was a logical baseline to compare against. And following on some of the shortcomings of this system, we developed a novel syntactically-guided bidirectional LSTM model, which processes basically a walk through a dependency parse of the input. Both models are trained using Hugging Face's trainer class with early stopping-based hyperparameter search, and in the case of the LSTM, we also search for architectural configurations. So briefly, this is our BioBERT baseline, which is simply a BERT sequence classifier where the sequence has been edited to mask the drug and protein phrases with special tokens. Here we use chem and gene for historical reasons, but you can use any arbitrary pair of tokens. And the entire BERT network is then updated to classify the special CLS token, the blue token here, with one of the 13 interactions or the negative or null interaction. We noticed, however, that the BioBERT classifier performance began to drop as the distance between the drug and the protein increased. And this inspired our use of a syntactic parser, which is sort of the go-to way of accounting for long-distance grammatical relationships between words and phrases. So it inspired our BiLSTM model, where the word piece token activations are averaged to produce BERT-derived encodings for true syntactic tokens in a dependency parse. So here we use spaCy for dependency parser. We then find the shortest path between any token in the drug phrase and any token in the protein phrase, and we enumerate them along that path in the parse that connects them. This sequence of token encodings and a few other parse-derived features is then fed through a bidirectional LSTM model, which classifies the concatenation of the final hidden activations for the forward and backward directions. Crucially, though, we did not fine-tune the underlying BERT model using the BiLSTM error gradients, but doing so would likely improve performance. What we see now is that this preserved the close-range performance of the BioBERT model while improving long-range performance as the drug and the proteins get farther apart. So here briefly is the aggregate performance metrics of accuracy, binary accuracy, which is basically detecting whether or not there's an interaction at all, precision recall, and F1. And we see that the BiLSTM outperforms the BioBERT baseline in all areas except recall on the test set, and it outperforms the DrugProt mean performance in all areas except recall on the test set. So one possible reason for the lower recall score is the added noise from having an imperfect parser in the loop. If the parse goes awry, then it's not going to find the same relationships that it found in training at test time, and therefore it'll have lower recall, but higher precision. Here again briefly is the per category precision recall in F1. So of note is the poor performance on low data types such as agonist inhibitor, and these data types are also quite complicated interactions that would require more data to be correctly categorized. So we plotted the training set size versus precision recall in F1 on a held out dev test set, and also F1 on the remainder of the development set. And we see that performance basically tracks with the training set size with four notable exceptions. There's the agonist and antagonist categories which outperform expectations, probably due to the fact that they're lexically cued. So there's always a word like agonist or antagonist in 96 to 97% of the cases. And then there's the inhibitor and indirect downregulator classes which underperform expectations. Looking at the data, we found them just to be quite complicated sentences, and also those two are often conflated with one another, which is understandable because they're similar interaction types. So for future work, we'd like to first of all, update the underlying BERT network in the Syn-BiLSTM model. We expect that'll improve performance. We'd also like to perform whole abstract reasoning by propagating the most confident interaction to all instances of drug-protein pairs throughout an abstract. This would require named entity resolution across instances, though. And finally, we'd like to have rules to handle these low data cases like agonist inhibitor to recover from unreliable statistical predictions. Thank you very much.